Good day, everyone. We're reviewing Wetzel's Limnology textbook. This is edition number 4, and we are focusing on chapter 9 today, Structure and Productivity of Aquatic Ecosystems. Interactions between species, especially predation and competition, have long been studied by community ecologists. Predators are known to be able to control the densities of prey in some ecosystems, establishing a trophic cascade. Trophic cascades occur when predation controls the density of prey, who themselves are controlling the density of their prey, which is then released from predation pressure. Trophic cascades are known to occur in both rivers and lakes. Often, the top predator is a fish that keeps the density of an algal grazing prey species low. As a result, algal growth is not consumed, and standing stocks of algae increase. Trophic cascades may have more than 3 levels and may occur only at particular times or places. For example, they may only occur once the population size of algal grazers is large enough to control algal biomass, or only on certain substratum types. Interspecific competition occurs in freshwater ecosystems. Competition, predation, and other interactions between species are vulnerable to decoupling as species phenologies are altered by climate change. There remains considerable uncertainty around what this will mean for ecosystems in the future, but the development of novel ecosystems is one possibility. Many community ecologists focus on whether environmental filtering or niche partitioning control assemblage composition. The concept of environmental filtering is that species present at the site will be those that have successfully passed through the filter of environmental conditions at that site, as determined by their species traits. Only once species have arrived at the site can local factors influence community composition. This theory has been criticized for ignoring the role of species dispersal capacity and landscape connectivity in determining which species can arrive at a site. Freshwater species face greater dispersal challenges than do marine or terrestrial species because freshwater bodies are often isolated from each other by terrestrial landscapes. Human interactions often increase this isolation by removing habitat. Also, the restoration of high-quality habitat has shown that dispersal limitation influences community composition. Both dispersal and environmental filtering may influence community composition, but their relative roles at any particular water body or ecosystem type will vary. Where environmental filtering exerts strong pressure or on communities, the convergence of species traits may occur. For example, microbes in hot springs are limited to extremophiles that tolerate or need very high water temperatures. Those types of communities tend to be relatively low in diversity. Where trait convergence occurs, species may be regarded as occupying similar niches. The concept of the niche has been criticized and defined in various ways, and there is insufficient space to encompass that debate here. But for our purposes, the niche can be defined as a suite of environmental requirements and tolerances of a species. Niche partitioning describes how species with similar niches divide up habitat in space and time to coexist. Other evolutionary timescales, competition should create selection pressure for species differentiates. Term niche differentiation. This is a fundamental way in which evolution creates biodiversity. However, competition between species must affect sexual selection, or at least select individuals prior to reproduction, for evolution to cause differentiation. Studies in freshwater show that both niche partitioning and environmental filtering occur. The pressure exerted by environmental filters varies in space and time, and this means that the pressure on similar species to diverge will also vary, potentially enabling similar species to coexist much of the time or to coexist in different habitat patches within an ecosystem. How the freshwater biota form communities with particular structure and composition is still the subject of research. However, the variables important to different parts of the biota are now well understood for many groups and are described in subsequent chapters. Overall, it is important to remember the main factors that structure communities: environmental conditions, species traits, biotic interactions, and landscape connectivity. Of these, the effect of global warming may alter environmental conditions and landscape connectivity, influences biotic interaction. Importantly, how species respond to these changes will be determined by whether their species traits enables persistence, either in situ or through dispersal to cooler location. Meta-communities dynamics. Meta-communities are communities connected by the dispersal of individuals among them, analogous to meta-population. They are often conceptualized as a cluster of lakes, each of which provides a range of environmental conditions and community composition, a subset of which supports metapopulation of multiple species. The dynamics of meta-communities are therefore reliant on dispersal and connectivity among the communities in the metapopulations. Most lake rivers and river reaches are now regarded as being part of a meta-community connected by dispersal. Restoration of one lake within a meta-community will see recolonization of the restored lake by species capable of dispersal among lakes, provided there are no barriers to that dispersal. Similarly, a river reach containing a series of riffles, pools, and runs will be connected by dispersal to other reaches. Species that prefer minimal flow will form metapopulations among the pool within their dispersal range, and together these metapopulations form a meta-community. Rheophilic species that live in riffles will also form metapopulation and a meta-community. This understanding of community dynamics across larger spatial scales has changed the way freshwater ecosystems are managed. It is now regarded as essential that lakes are managed in groups rather than as individual water bodies, and that connectivity along rivers is important to maintaining ecosystem function and river biodiversity. The meta-ecosystem concept. The meta-ecosystem concept recognizes that ecosystems are connected by flows of organisms, energy, and materials across ecosystem boundaries. It progresses beyond the RCC, which is the river continuum concept, because it reviews riverscapes as networks of rivers connected by flows of genes, species, materials, and energy in multiple directions. It thus moves away from the predominantly longitudinal downstream flows encapsulated by the RCC. Atlantic systems such as lakes and ponds are also part of this meta-ecosystem because they are connected through rivers and groundwater flows, but also through animal movements or passive, wind-driven dispersal. Diversity and diversity metrics. Diversity may be simply described as a number of species living in a defined area, which can be anything from a single site or water body or scaled upward to the size of a continent. When diversity is determined at the habitat level, it is referred to as alpha diversity, whereas diversity at the landscape or regional level is referred to as gamma diversity. Species turnover between different habitat patches within the landscape is defined as beta diversity. This is an intuitively easy concept, but in practice, measuring that diversity can be difficult. Many species of microbes, algae, and invertebrates have not been described or named, and thus may be difficult to identify and quantify. Measures of diversity rely on a species concept, but this itself can be difficult to apply to microbes. There are many ways to quantify diversity. Species richness is the count of the number of species in a defined area, or volume or amount of sampling effort, and is often used to quantify diversity. If similar sampling methods are used, species richness can be easily compared between locations. As species richness estimates are heavily influenced by sampling efforts, species saturation or accumulation curves are often used to assess whether sufficient volume or area has been sampled to provide a representative estimate of species richness. Only when the curve reaches an asymptote can the sample be considered sufficient for reliable quantification of richness. These species accumulation curves also allow researchers to compare species richness across different populations. However, anyone who has studied an ecological community will soon see that some species are common while others are rare, and others lie somewhere in between. Species, therefore, differ in their abundance or density. Relative abundance is often used to compare species. It is defined as the abundance of each species collected using the same sampling method. Relative abundance can be very useful because it facilitates comparison between, whereas absolute abundances are difficult to estimate for most species and are often less useful. Species evenness considers the number of species and the relative abundance in a certain area and is often expressed as the Shannon-Wiener index or the Simpson index. Therefore, although we might measure species richness, the species counted will occur at a wide range at different abundances. What does this mean and how does it affect how we quantify diversity? There are two well-known relationships that affect species richness and therefore affect how diversity is quantified: the species area relationship and the species abundance relationship. As the area or volume of habitat increases, the number of species encountered will increase. This is the species area relationship. For this reason, when comparing different places, researchers endeavor to use the same size and number of samples so that the probability of encountering species is the same in each place, and thus species richness can be compared. Similarly, as the total number of individuals collected increases, the probability of encountering species will increase. This is the species abundance relationship. Where two places are compared and one has higher abundance, it will likely also have more species. Thus, when comparing species richness between places, researchers often also measure abundance. For these relationships, rarefaction can be used to estimate species richness either at equal numbers of samples or equal abundance of individuals. Rarefaction is a statistical technique that randomly resamples species from a dataset to show the relationship between the average number of species found in each of these random samples and the number of samples or individuals. Rarefaction curves usually show a steep increase at first when every new sample results in a large number of new species found, and then reaches an asymptote when additional samples or individuals only results in the identification of a few additional rare species. Rarefied estimates therefore provide more comparable measures of species richness as they take sampling efforts into account. Differing abundances of individuals in different places may be a simple product of the amount of energy and nutrients available in that ecosystem. For example, lakes that receive excess nitrogen and phosphorus from agricultural activities in their catchments often have high algal productivity and density. This provides a large food resource for animals, which can also reach very high densities. However, in such situation, diversity often declines because environmental conditions are only suitable for a narrow range of species. Where the ecosystem to be compared have apparently similar levels of productivity and the same sampling effort is used to collect data, simple species richness is often readily comparable. Species richness provides information on the number of taxa present at a time and place. Where those taxa are based on reliable phylogenies, phylogeny describes the evolutionary relationships among taxa, it can also be used to measure phylogenetic richness. With modern molecular genetics methods, phylogenies can be constructed through genetic analysis of the species collected. This may explain why some locations have a greater number of species within some taxa than others. For example, comparison of taxonomic richness made at a large scale, at large spatial scales, for example continents, will in part have been determined by the evolutionary history of those lands. Phylogenetic diversity can distinguish between historical contingency and contemporary differences in richness that can tell us about factors currently affecting diversity. Molecular genetics may also provide a better description of species richness than taxonomic studies if there are many cryptic species present. Cryptic species are hidden within population as they belong to two or more species that appear morphologically identical but are genetically distinct. Difference between species' physical appearance can be very subtle, yet animals and plants may perceive these differences such that they only breed with genetically compatible individuals. Cryptic species are often identified among aquatic plants and animals, sometimes in surprising numbers, and morphological, physical, or behavioral differences between those species are often subsequently observed. One recent way to measure richness is to use environmental DNA. Each animal, plant, or microbe living in freshwater will shed DNA into that water, and eDNA, or environmental DNA methods, then can sequence those genetic materials. A list of gene sequences is then compared with libraries such as GeneBank, so that names can be put against each sequence. In much of the world where species are poorly known, eDNA provides a long list of species against which it is impossible to put species names. In better studied parts of the world, eDNA can provide an accurate list of species present in a water body at the time of sampling, including cryptic species and those that are secretive and hard to sample. eDNA is therefore an increasingly popular way to measure diversity in freshwaters. Another way in which ecologists often measure diversity is through functional or trait diversity. Functional groups are groupings of species that fulfill similar function in ecosystems. Trophic groups such as predators and grazers, or carnivores, herbivores, and omnivores are examples of functional groups. Food webs and ecosystems may differ in functional diversity. Species traits are heritable characteristics that determine how species interact with their environment and each other. They include variables such as body size, methods of respiration, and dispersal methods. Species may be assigned to different combinations of traits within a matrix, and then differences observed when ecosystems are compared. For example, shallow, highly vegetated lakes may show marked diel fluctuation in dissolved oxygens due to high levels of photosynthesis during the day and equally high respiration at night. Aquatic animals present in those lakes may be predominantly air breathers. For example, turtle, frogs, etc., for whom low dissolved oxygen is irrelevant, and gill breathing species may be quite rare. Aquatic animals present in those lakes may be aquatic animals present in those lakes may be predominantly air breathers, for whom low dissolved oxygen is irrelevant, and gill breathing species may be quite rare. Thus, the composition of species traits present within an ecosystem can often be related to environmental conditions or the impacts. Thus, the composition of species traits present within an ecosystem can often be related to environmental conditions or the impacts of disturbances to those conditions. In summary, or Chapter 9, an ecosystem comprises all the life forms and the inorganic context that they live in. A catchment is the area of land surrounding a water body through and over which water flows into that water body. Human effects on global climate and elements cycling are now so profound that we live in a potentially new era, the Anthropocene. In flowing or low-tech waters, we refer to the water regime as the flow regime. This is because the unidirectional nature of flow rivers and streams distinguishing them from other ecosystems. In lentic or standing waters, we often refer to the water regime as the water balance. A lake is separated into the open water pelagic zone and the littoral zone, the latter consisting of the bottom of the lake being basin colonized by rooted microphytes. The sediment free of vegetation that lie below the pelagic zone, are referred to as the profundal zone. The littoral profundal zone is the transitional area of the sediment between the littoral and profundal zones, occupied by scattered benthic algae or microbial mat. In rivers, the riparian zone refers to the riverbanks and their vegetation, which can tolerate periods of inundation during floods. River segments are length divided by tributary junction, and nested within these are reaches. Nested within reaches are habitats such as pools, runs, and riffles. As water moves through the catchment and fringing zone into a water body, the type of nutrients and OM, or organic matter, in the water and their concentration are modified. Autochthonous production means carbon fixed within a water body. Allochthonous productivity arises in the water body through terrestrial subsidy from terrestrial or fringing plants. In order to evaluate the productivity of a system, it is important to realize that production is a rate and often expressed as a units of mass or energy production per volume per time. In self-sustaining biological communities, functionally similar organisms can be grouped into a series of operational levels of producers and consumers, together constituting a food web. Since organisms expend a considerable amount of energy for maintenance, only a portion of the energy at one trophic level is available for transfer and used by higher trophic levels. A population comprises a subgroup of a single species that are interbreeding. Ecologists use molecular genetics to define the spatial and temporal extent of aquatic populations. The area that a population occupies is determined by three variables: the dispersal ability of the species, the distribution of suitable habitat within the surrounding area where the species, and the connectivity permitted by the landscape. Phenology is the time at which important life cycle events occur for a particular species. A metapopulation is a group of patches occupied by a subpopulation and connected by the dispersal of individuals among the patches where interbreeding occurs. A community refers to a group of interacting populations of different species. How the freshwater biota form communities with particular structure and composition is still the subject of research. Overall, it is important to remember that the main factors that structure communities are environmental conditions, species traits, biotic interactions, and landscape connectivity. Meta-communities are communities connected by the dispersal of individuals among them, analogous to metapopulations. The dynamics of meta-communities are therefore reliant on dispersal and connectivity among the subcommunities in the metapopulation. The meta-ecosystem concept recognizes that ecosystems are connected by flows of organisms, energy, and materials across ecosystem boundaries. Diversity may be simply described as the number of species living in a defined area, which can be anything from a single site or water body up to a continent. And that's going to be it. And that's pretty much the end of chapter 9. And I will see you tomorrow, and we'll start on chapter 10. And chapter 10 is the water as a chemical environment. Have a good day, and I'll talk to you soon. Bye.